Hello everyone, this is the Chinese Mitten Crabs aquaponics project, and today we're going to talk about photosynthesis. First, let's talk about the basic concepts of photosynthesis. Plants and algae are autotrophs. They make their own food by photosynthesis. Photosynthesis is the process by which organic substances, such as carbohydrates, are made from simple inorganic substances, carbon dioxide and water, using light energy. During the process, light energy is captured by chlorophyll in plants and algae. Chlorophyll is found in chloroplasts in most plants. The light energy captured is converted to chemical energy that is stored in the carbohydrates produced. Oxygen is released as a byproduct. The overall equation for the process of photosynthesis is carbon dioxide added with water with light captured by chlorophyll turns into carbohydrates and oxygen. Photosynthesis is an anabolic process because it involves the synthesis of complex organic molecules, carbohydrates, from simple molecules, carbon dioxide and water. Proteins and lipids are made from intermediates of photosynthetic reactions. Their production requires minerals absorbed from the soil. From these organic nutrients, plants and algae can make other materials they need. Let's now talk about the site of photosynthesis. In plants, leaves are the main site of photosynthesis. There are many cells containing chloroplasts in leaves. Both of the leaves and chloroplasts have structural adaptations to serve their functions in photosynthesis. So what are the adaptive features of leaves for photosynthesis? The morphology of the leaf and the distribution of chloroplasts in the leaf are well adapted for photosynthesis. From top to bottom, the leaf is covered by a cuticle. Cuticle, sometimes known as the waxy cuticle, is a layer of wax that is waterproof and prevents water from diffusing to the upper epidermis. Then, it is the upper epidermis. Except from the guard cells, most epidermal cells have no chloroplasts. The following layer is the palisade mesophyll, made up of tightly packed cells containing many chloroplasts. This is the layer which most of the photosynthesis takes place. Next down is the spongy mesophyll, made up of cells in irregular shapes. The cell contains fewer chloroplasts. This layer also contains air spaces for gas exchange. Within the spongy mesophyll are the vascular bundle, xylem and phloem. Then it is the lower epidermis. It usually contains more guard cells than the upper epidermis. Last, another layer of cuticle. This is a table showing how the structures of a dicotyledonous leaf is adapted for photosynthesis. First, the leaf blade is broad and flat, which provides a large surface area for absorbing sunlight. It is also thin, so that gases and light can reach the photosynthetic cells easily. Secondly, the palisade mesophyll consists of tightly packed cells that contain many chloroplasts located on the upper side of the leaf, exposed directly to the sunlight. This allows effective absorption of sunlight. Third, the spongy mesophyll consists of loosely packed cells with many air spaces. The cells contains chloroplasts, fewer than the palisade mesophyll. This allows gases to diffuse freely inside the leaf and allows absorption of sunlight. Last but not least, the upper and lower epidermis is covered by a thin layer of waxy cuticle. This can reduce water loss from the leaf, so that water is kept inside for photosynthesis and keeping the cells turgid. Furthermore, it contains numerous stomata, especially on the lower epidermis. Each of the stoma is surrounded by a pair of guard cells containing chloroplasts. They are adapted to photosynthesis because stomata only opens when conditions are favorable for photosynthesis. They close when the conditions are less favorable. This regulates the passage of gases and water vapor into and out of the leaf. Last but not least, the midrib and veins. They contain vascular bundles which are made up of xylem and phloem. They allow efficient transport of materials, for example water and food, into and away from the leaf, and it supports and spreads the leaf blade. So what are the adaptive features of chloroplasts for photosynthesis? A chloroplast is bounded by a double membrane and is filled with a jelly-like fluid called stroma. The stroma contains enzymes that catalyze photosynthetic reactions. It also holds starch grains which are temporarily stored as photosynthetic products. A network of disc-like membranous sacs called thylakoids is suspended in the stroma. The thylakoids are arranged in stacks called grana. Chlorophyll is located on the thylakoid membranes. A chloroplast has about 50 grana, each consisting of about 50 thylakoids. This provides a large surface area to pack more chlorophyll for absorbing light. The grana are interconnected by extensions from the thylakoids. This allows efficient transport of photosynthetic products within the chloroplasts. After learning the structure of the chloroplasts, let's learn about the requirements for photosynthesis. We will study the factors required for photosynthesis. In each of the investigations, we need to determine if photosynthesis has occurred in the presence or absence of the factor studied. First, how do we know if photosynthesis has occurred? We can detect the presence of starch to test for the presence of photosynthesis. Glucose molecules formed in photosynthesis are quickly built into starch molecules. Therefore, we can carry out an iodine test to test for the presence of starch in a plant to find out if photosynthesis has occurred. We can detect starch produced in photosynthesis using the iodine test. First, pick a green leaf from a dicotyledonous plant that has been exposed to bright light for several hours. Second, boil the leaf in a beaker of water for about a minute. Then, turn off the Bunsen burner. Transfer the leaf to a boiling tube half filled with alcohol. Put the tube into a beaker of hot water. When the leaf has lost its green color, meaning it's decolorized, transfer it from the tube to hot water again. Spread out the leaf on a white tile. Add a few drops of iodine solution onto its surface. Observe any color change in the leaf. The iodine solution should turn blue-black all over the leaf. This indicates that starch is present in the whole leaf. We can detect the release of oxygen to test for the presence of photosynthesis. Oxygen is a byproduct of photosynthesis. We can also test for it in the gas produced by a plant using a glowing splint to find out if photosynthesis has occurred. First, set up the apparatus as shown. Dilute sodium hydrogen carbonate solution provides a carbon dioxide to the hydrilla. Secondly, expose the setup to bright light for at least 24 hours. Third, remove the test tube carefully and stopper the mouth of the test tube under water. Last, put a glowing splint over the mouth of the test tube to test for oxygen. The glowing splint should relight. This shows the gas released from the hydrilla is oxygen. Therefore, let's move on to the investigation of the requirements for photosynthesis. We will study the need for chlorophyll, carbon dioxide, and light in plants during photosynthesis. There are two setups in each practical. One includes the factor to be tested, which is the experimental setup, while the other does not, which is the controlled setup. If photosynthesis takes place in experimental setup but not in the control, we can say that the factor being tested is needed for photosynthesis. When we do photosynthetic experiments, we need to distarch the plant being experimented on. If we plan to use the presence of starch in leaves as evidence that photosynthesis has occurred, any starch that is present in the leaves before the investigation must be removed. Otherwise, we cannot be sure whether the starch detected at the end of the investigation is made during the investigation or not. Plants used in such investigations must first be put in the dark for at least 24 hours to remove all the starch in the leaves. In the darkness, the starch in leaves is converted to sucrose. Sucrose is then transported out of the leaves. This process of removing starch is called destarching. After destarching, we can investigate if chlorophyll is required for photosynthesis. It is impossible to remove chlorophyll from a green leaf without killing it. Therefore, we cannot study the need for chlorophyll by removing it. Instead, we can carry out investigation on plants with variegated leaf. For example, coleus. Chlorophyll is absent in the non-green parts of the leaves. The part of the leaf with chlorophyll is the experimental setup, which is the one with the factor being tested, while the part without chlorophyll is the control setup, which is the one without the factor being tested. On a side note, why do leaves turn red in autumn? Chlorophyll is a pigment that absorbs red and blue lights in the visible spectrum and reflects green light. Therefore, most leaves are green in color. In addition to chlorophyll, leaves contain other kinds of pigments. One of them is carotene. It absorbs blue light and reflects red and yellow lights. The light energy captured by carotene is transferred to the chlorophyll for photosynthesis. In summer, the abundant chlorophyll in leaves masks the color of carotene. In autumn, the chlorophyll in the leaves break down due to low temperatures and bright sunlight. The color of carotene appears, and therefore leaves turn red or yellow. Carbon dioxide can also be investigated if it is needed for photosynthesis. The investigation can be carried out by exposing a leaf of a plant to normal air, the experimental setup, and expose another leaf of the same plant to air without carbon dioxide, the controlled setup. Potassium hydroxide is used to remove carbon dioxide from the air inside the control setup. First, destarch a plant by putting it in the dark for at least 24 hours. Pick a leaf and test for starch. Secondly, set up the apparatus as shown. Third, expose the whole plant to bright light for about 5 hours. Then, test the leaves A and B for starch. Starch is present in leaf A but absent in leaf B. As leaf B is exposed to air without carbon dioxide, the results shown that carbon dioxide is required for photosynthesis. Another factor that can be investigated is light. We will expose parts of a leaf to sunlight, the experimental setup, and mask certain parts of the leaf, the control setup, to study the need for light in photosynthesis. A piece of black paper or aluminium foil can be used for masking. First, we can destarch a plant by putting it in the dark for at least 24 hours. Pick a leaf and test for its starch. Then, cut out 2 pieces of black paper of a similar size with a pair of scissors. Punch a hole at the center of each piece of black paper. Afterwards, attach pieces of paper to both sides of a leaf on a plant to fix them with paper clips. Then, draw a sketch of the leaf to show which part of the leaf is masked and which part is not. The plant is then exposed to bright light for 5 hours. Afterwards, remove the paper and test the leaf for starch. Draw another sketch of the leaf to show the result of the iodine test. The iodine solution turns blue-black in the leaf except for the masked part. This indicates that starch is not present in the masked part. The results show that light is required for photosynthesis. Water is also required for photosynthesis, but it is difficult to carry out investigations of the need for water in photosynthesis because water is needed for many other vital processes that support life in plants. Without water, the plant will die. This is the end of the video. Thank you for watching. Please give us a like and subscribe to our channel. Click on the tab to watch the next part of the video series.